Okay, now back close to livestock development. Animal production. Now closer, okay? I have to move a little bit faster now. We have to see what factors can attribute to the efficient productivities of our animal production, right? So the feed resources that will be, consumed by the animals are very important among other important factors. Feed resources or nutrients that will be taken in by the animals. Now the resources are very diversified from the common one and the uncommon ones, okay, or conventional, okay, or crop residues, industrial crop byproducts, and the co-products from the industries. All of these all together, are essential for the potential productivity of the animal production. We have to be able to identify what has been there, what can be potentially utilized more, what can be utilized less, what could be distributing to better quality of the product. We should be able to know in order to have a chance to select and formulate the ration for species of animals you are engaging in, right? All of these need to be identified and need to be tailored down, okay? So we have to move on. After that, like in ruminants, particularly like this is what I've been working on. Ruminants, both large ruminants and small ruminants. Beef cattle, dairy cattle, buffaloes, okay, sheep and goats, all together are quite important. They are very efficient in utilizing the fibers, particularly the fibers, the rough fishes and fibers. And the fibers are the main feed resources, right? We all know that. But how can we effectively utilize this fiber? We have to see, like, for example, crop residues from rice, is the rice straw. Rice straw is abundantly available in Indonesia, in Thailand. So all year long we have rice straw available. If you can do like 3 times rice cultivation, there you go, you have lots of rice straw available. And rice straw is rich in fiber. This is an example. But you need to improve the quality of the rice straw. There are many methods that can be employed. Okay, there are many work in the literature. And may I say that during my postdoc work, I was able to be working with the most famous professor on straw improvement. He was Professor Frik Sundstol at the Agricultural University of Norway. So I was very lucky to be able to work for a 1-year postdoc there with him, and I learned a lot. And I adapted myself, and knowing many friends in Norway, and I can speak a little bit of Norwegian as well. Yeah, like good day, hvordan har du det? Hi, how are you? Feel tak. Fine, thank you. So just like that. Okay, the young people can do even more, right? Now you guys can do more. Okay, straw treatment is very important. That I have, come back. I have come back to Thailand, and started the improvement of rice straw, and many friends in Indonesia joined me, and we work together. Anyway, so many methods available. We have to explore and then employ them. What is the most suitable and what would be the yield, effectively of that methodology. So there are many things that you can explore and bring in. And after that, we have to fit well into the feeding systems. There are many feeding systems, like in our countries. The dean just said that, Indonesia grazing openly or tethering or like housing, under the house or near the house. These are very diversified, and we have to identify them. What would be what's existing, and we have to tailor it down to such a system that will be best benefited into the system in terms of the resources of feed and also in terms of nutrient utilization, and for the goals of the productivity. What were they? What are they going to be, right? In order to best effectively utilize the system and the classes of feed resources down to the nutrients. Okay, all of this together have to be identified for the efficient ruminant production in the future, especially now. Smart farming or precision farming have been addressed very strongly, and the young people, as well as senior research, we have to learn, right? We have to see what can be done and what we can add on into the productivity, the methodology, in order to empower all the system together like that. So we have to work together. At the end, we will be able to obtain the efficient production. Now, closer to ruminants, even more. Even more. As stated very strongly, very loudly, we heard of this, feeding the bucks, feeding the cows. What it means? It means that the cows will not, utilize the nutrient, by themselves directly, but they have to go through, process of degrading and digest degraded and absorption later on. Okay, degraded by the microbiome available in the rumen, which is very important. Microbiome, microbiology, microorganisms available, which is anaerobic fermentation. It's very important here because the fermentation vat, without fermentation vat here, the animal would not yield the nutrients that they should be, right? So you can see this picture here. The rumen, where the microbes, particularly bacteria, protozoan, fungi, they live there, and they live in consortium, and they work together, synergistically, and also sometimes like protozoa cannot be all good, right? For example, we have to definite them out in order to reduce the population of protozoa. To reduce the population of methanogens. This is a good example. We have to see what would be good and what would be reduced for the betterment of the rumen. But most importantly, the rumen needs to be healthy. They need to be in a good condition, particularly the rumen pH. Rumen pH needs to be in the range of the normal range of like 6.1 to 6.7. Okay, below and above will be some potential incidents to the ruminants would occur. So we have to identify them and to be able to maintain, and also to, at the same time, to increase the productivity of the rumen fermentation in order to prevent these incidences like bloat or acidosis, right? And to reduce the global warming gas, carbon dioxide, particularly the methane, emitted out from the rumen into the atmosphere, which will then eventually impact on the global warming. We need to reduce them here. But to reduce the methane or carbon and hydrogen in rumen is the way for increasing the efficiency of the production. Okay, because you will be able to trap the carbon and the hydrogen here for the synthesis of the products that will be in the form of meat or milk later on. So to talk about mitigation of methane is not per se to reduce it down only, but it means that it will potentially increase the efficiency of production activities by means of having more meat, quality meat, having more milk, quality milk, and more financial return. This is what the farmers need to hear about mitigation of methane. We cannot go down there, my friends, staff members there, to the farmer and say, I like to help you reduce the methane, right? They would not be interested in knowing that, but they would be interested, on the other hand, how much money, how much product I would gain from this, I would get from this, and how much more money I receive from selling my meat and my milk, right? So we have to gradually explain to them that, okay, reduce the carbon, reduce the hydrogen, means that more meat and more milk will be gradually enhanced, and eventually the budget that they will receive. This is a very important message. We've been having problems talking about in developing countries that while methane mitigation transfer cannot be possible or cannot be successful. This is the message I like to transfer to all of you guys, that we need to transfer that, apply it into the productivity of the animals and the economics, okay? You can see here, theoretically, you will see here, this is the intake of the ruminant is very important, especially intake of the fibers, intake of the poor quality roughages is very important here. Intake or consumption of the roughages is not sufficiently consumed or obtained by the animal because of quality. Because the quality of the feeds have been very low, right? Quality of particularly the crop residues like rice straw or sugarcane top and so on and so forth have been very low, and even lower than 50% of TDN. Okay, if lower than 50, 55 here, 55 and 50, because the rumen will be filled already. The animal cannot eat more, and the uptake of nutrient would be very low already. So we need to improve the quality of the roughages, okay, in order to obtain more intake and more nutrients, okay, and these nutrients will be gradually be degraded and absorbed for the synthesis of the products, okay? So bear in mind that we cannot say that we are unfortunate, right, in developing countries or in the tropics to have this, but in a way I would say that we are lucky to be able to have these crop residues, the biomass available throughout the year. We need to know how to utilize them effectively, wisely, in order to get the nutrient even more from this biomass, okay? See here, you see there's a feed when coming into the rumen here. You can see all of them will be degraded, okay, by the enzymes created by the microbiome here. This is an area of interest by scientists right now, that like graduate students, postdoc can work more on the microbiome, from bacteria, protozoa, fungi, and methanogens. You can see what's available there, how they work, how they degraded biochemically, microbiology, microbiology, right? Tools in order to degrade by their enzymes. What would be the effective way to do that in order to get the products here? Fermentation products like from volatile fatty acids, right? You are aware of this, okay? And the microbial protein synthesis from the nitrogen.
Recording stopped.
Especially ammonia here for the lower gut. All of this would go to the lower gut, except the volatile fatty acids, right? That we absorb through the rumen of.
Recording in progress.
Of the ruminants, right? So this is very important. That's why I bring this up for the understanding of the students to study more and work more with this. Okay, as you can hear that the work has been reported by Hungate 1926 that has a great book that we have used a lot, and many papers that have been published dealing with the rumen microbes. If you go after this, you can get lots of information. Out of that, I like to address one of the papers that published, which is very significant and very knowledgeable. That is the paper by Russell and Richlick, 2001, from Cornell University. That I recommend you to read more in order to equip yourself with more information, right? Okay, let me go on. Now, to the methane, especially enteric fermentation here, I need to go a little bit faster, I suppose, right? So here, the overall methane production has been attributed by many sectors, firstly by agriculture. Okay, by agriculture as a whole. It has been reported by Steinfeld from FAO long time, and Goodland and Anyang also reported later on, right? So you may have heard of these names that, okay, agriculture could contribute up to like 30% of total global warming gas, and in the 30% here, the livestock enteric fermentation could be accounted up to like 20%. Okay, 20 to 30% of the 30%. So it varies from the production system, it varies from the feeding system, it varies from many factors, okay, from this. But most importantly is that methane production from enteric fermentation is from ruminants. We cannot go away from it. So we have been aware of, and we should be able to help mitigate, okay, the potential methane gas production emitted out into the atmosphere. Here the cow is here. The cow has been like, giving bad news, right? Like, okay, cows, ruminants, sheep, and goats are the bad guys, right? But actually they are not. They are partially producing the methane. And the methane is, emitted out into the atmosphere, CH4 methane. And this CH4 has a long life of about 10 years. And that will be regenerated into the system. It's just like carbon or hydrogen from the fossil fuels, which has a long life cycle. Okay, whereas the methane, the carbon and the hydrogen from cows, from sheep and goat, would be much shorter, because they will be coming back into the system, especially into the feed system of the feed resources of roughages, that the carbon will be bring back into the biomass of grass and by photosynthesis. Okay, and the grass will be consumed by the ruminants, right, as feed, and will be degraded in the ruminant by the microbes. There you go. You can see that, there is not everything is all bad, right? It is there, but it is coming back shorter than from the fossil fuels, which is different, significantly different. May I just have a sip of tea? Now you can see that. So there are ways out. We can help through the fermentation efficiency, as I have touched a little bit there. Through the fermentation efficiency by using the carbon and the hydrogen in the hydrogen sink into the system to produce more volatile fatty acids, particularly the propionic acid that would be used for as the to generate glucose, to gluconeogenesis in the liver, right? So that is very important here. So we can fight against it that okay, it's there, but it's partially, but it can be more efficient if we can trap it and change it in a different form. And you can see here the methane produced from different classes, from different sources. You can see here, this is enteric fermentation of about, I said about 30%, right? It says 30 to 40%, right? So this is 39%, okay, which is among the volume that produced as a whole. Okay, here we go. It is biogenic carbon cycle. I brought out that methane from emitted out by the cow here is a biogenic carbon. It's not like fossil carbon that is also can be useful in recycling back to be used like after 10 years, right? It's not after 1 day or 1 week or 1 month, but it's after 10 years. The life cycle is much shorter. Okay, we can, but I don't want all of you to think that, okay, we just wait like that, fight for it like that. No, we need to improve the fermentation, you know, in the immediate way here for the more efficient utilization of the carbon and hydrogen over here through this fermentation here. Okay, this one here, you can see that this is through the succinic acid. The hydrogen produced, you know, rather than synthesizing into the methane here, into the methane, right? Into the methane right here. So the hydrogen will be synthesized to methane here. We block it up. We block it up here in the rumen here. We bring in the hydrogen here through the succinic acid cycle to produce propionic acid or C3. Okay, this is the common randomization pathway in the rumen. It's more effective. It's more efficient. Okay, so we have ways to do that, especially through dietary manipulation. There are many ways of reducing methane. Okay, among those methods, dietary manipulation is one of the promising ways to reduce methane. Here, back into the rumen, the late Professor Oscorp, whom you guys know him very well, is a worldwide scientist, okay, from Scotland, and he has been working out in Asia for many years, and especially in Indonesia. He has been working in many universities there. You know, we all love him, but unfortunately, he has gone now. Okay, he has done a great job, and I learned a lot. You know, I had a chance to visit him long time, and you know, in his place, in his house, his farm in Scotland. You know, he's a great guy, great scientist. Okay, so he showed that okay, fermentation patterns that will be dependable or impacted by level of roughages to concentrate feed ration. You can see, okay, more roughage, you get more acetic acid. Okay, if you reduce that, you get more propionic acid. Okay, like that. And our work also follow Wanapat here at all. Okay, here follow similar pattern of the work of Professor Bob Oscorp. Okay, this is very good pattern. We have to be able to look at how it works in the rumen. With this fermentation end products, it will greatly impact on the pH, on the rumen pH. We need to buffer the rumen pH not to be less than 6, or if you can, 6.1 or 6. You know, if you need to go down, but you have to buffer it up to 6.3, 6.4 in order to have a common fermentation, okay, and to avail the microbiome here to work even better for the fermentation end product through manipulation of the feed ration, the ratio, for example, ratio of roughages to concentrate, okay, where would be most suitable, where it would likely to be efficient. Okay, the ratio has been shown that the common ratio, okay, is like 50 to 50 or 60 to 40. But if you want to move up to fattening scheme, fattening regimes, you may have to lower the roughage down to like 40 or 30 to 60 and 70 concentrate, things like that. It depends on the production systems and also the production goal of your production system, what you aim for, what would you decide to use, which ration. So you see this one here, this is the book, the PowerPoint from my friend Professor Edward from New Zealand, and lent me this very nice fermentation in a rumen that will yield the methane. This methanogenesis is quite complex and requires methanogens during the reduction processes and to secrete complex enzymes and the cofactors, okay, in the system, and eventually they yield the methane, and we have lost the carbon and hydrogen through methane. So we need to capture the carbon and hydrogen over here through the synthesis of C3, as I have just addressed. Okay. May I just skip this one? Okay. I skip this one. This is the methods for improvement of the crop residue, especially like rice straw. There are many methods: physical method, chemical, physiochemical, and the biological, all the combination. So you should explore this, what can be done. But mainly here in Europe, they use a lot of chemical. Where during my time, a long time for postdoc, I used the chemical method, okay, especially the ammonia treatment that has been very popular in Norway and Denmark and Sweden during that time. Anyway, you can search back and see, okay? Two of my papers also published in Animal Feed Science and Technology during those years. Okay, now, so you can see here, rice straw, Fricksunstol, and other method. Okay, other method can be searched for in order to loosen the bonding or the lignocellulosic bonding of the fiber, okay, in order for the enzyme to easily attack the bonding, and also the ability to dissolve the compounds, the soluble compound that may be that will be released out in the rumen, okay? So just in short, that the treatment will render the availability of the enzymes secreted out by the microbes, and the availability and the facilitation of the microbes to adhere for their activities. Okay, long time I show you that we started like long time. Okay, here we go. We work with the farmers, the dairy farmers in the Northeast, and they still depend on the rice straw. Rice straw is available now. It's loose. This one loose form, but later on it's bale form, and the bale form here is very costly right now, but it's well utilized by the ruminants, by beef and dairy, in particular dairy cattle throughout the year. Not only during the summertime, but throughout the year, because here you can store the rice straw bale, right, for later use, like that. This is simply done. It can be simply done in terms of urea treatment. We started with 5% urea treatment. Okay, there's many work from Indonesia as well. Okay, that have shown the positive impact of urea treatment. Okay, with the cost of urea treatment of urea, right? So we have been able to lower down the concentration of urea to 2%. By ways of lowering, we can add on with another chemical source. Okay, here we can use lime. Okay, lime, calcium hydroxide, lime, 2x2 in short, 2x2 urea lime. Okay, which can result in the efficient use of degraded of the straw in the rumen, like that, or you can build it up like that. Okay, I mean, this is another form of putting loose straw into the bale like that. You can, plastic wrap is becoming more important. And Indonesia, also I have seen that already, is widely practiced by the farmers now. You can see here, this one is in China. This one is practiced in China. Okay, now just some result of the straw treatment, 2 by 2. You can see 2% urea and 2% calcium hydroxide can significantly improve the digestibility of the nutrients as compared to the 5%, and the cost was significantly lower than using only 5% urea. Okay, the propionic acid and population of bacteria and improvement of the effective microbial nitrogen synthesis here have been significantly improved by urea treatment as compared to other treatments, as compared to the control, but similarly with the 5% urea. But the cost of treatment can be significantly lower. In milk yield, you can see that 5% here and 2 by 2% here was similar in the impact, but much higher than the control, untreated. So, to lower the concentration of urea from 5 to 2 should be recommended. And you can see in summary, you can see here how urea and lime can improve the straw treatment and utilization. Okay, they can improve the fermentation and onward to the intake of rice straw. As I have shown you, that intake is very important. That has been collected that has been obtained very low, but with the treatment could be much higher. Okay, now this is some treated straw color. You can see it's more brown. It's brown, dark color, as compared to the untreated here. Cover how long? 10 days. Just cover 10 days, not 21 days. To only 2 days, you can open up and fresh it and use it in a fresh form like that. No need for drying. Just adapt the animals, okay, and then feed it directly into the animals. Another feed, I go a little bit faster on this one. May I just bring up cassava, tapioca? You have a lot. You have a lot there. It is very potential. The root of cassava is there for energy source, carbohydrate, right? As a carbohydrate source, and you can see that, and you can say it can be degraded very rapidly in the rumen. Okay, but the biomass of the leaf and the stem, the young shoot, the young stem can be utilized as after sun dry. So a lot of work that I have done on cassava hair after drying, okay, can be utilized to use it as a source rich in protein. And by the way, cassava contains phytonutrients, especially those condensed tannins. So this is very important now, that condensed tannin in cassava leaf and stem, especially in the leaf, right, can be very beneficial for the microbes to be more effective in degrading the feeds, the nutrients, and to lower the methane production in the rumen through utilization of cassava dye, cassava leaf, or cassava hair. Especially, you can see that. And another form of making cassava, another way out, is to ensilage it, to make silage. See, this one is grown after 4 months, and we just cut right from the stem and chop it up and manually, make it into stems and store it in the drum like this. Like this one, it can hold up of like 50 kg each drum here, and see, you get beautiful silage of cassava. You can see the picture here. It's only like 10 to 14 days in silage and siling time, and that will end up having more quality, high quality of cassava silage that could be directly fed to sheep and goats, beef cattle, dairy cattle, beautifully. So you have to manage the biomass, okay, how you can do it. So you can benefit from the protein, which is quite high, 25% at least, and also the condensed tannin in here can be beneficially utilized in the silage. This is what we have in the topics. This is what we have in developing countries. We have to identify, explore it, and use it more. By use of the cassava top silage here in various forms, in various ratio, you can see that they could significantly improve the intake and the fermentation in products, especially the propionic acid. Okay. Another work in dairy cows, you can see using cassava silage here was significantly, lifted up the milk yield, 3%, 3.5% fat corrected milk here, significantly, you can see as compared to the control. So it's there on the farm, it's there at the gate. So you need to explore and then intervene feeding system like that. Using cassava, you can see here, you can see good pictures, beautifully, managed. If you can manage like that, you can harvest it and make hay or make silage, right? This picture shows hay mixed with the concentrate. This is a homemade concentrate, done by the farmers. And we're training the farmers to use chopped cassava hay as a part of protein to reduce soybean meal. There's no soybean meal used at all, but we use other sources also because of the price of soybean meal is skyrocketing. It's so high. It's improving. I mean, ruminants should use something else that is with a lower cost, okay? And then it could be more productive as well. You can say this is a good example of using locally available feed resources such as cassava hay, chopped cassava hay, and train them in to make homemade concentrate, farm-made concentrate. Make on farm. Farmers have choice. They don't have to buy commercial feed all the time. They can make their own concentrate through the training activities of students, of staff, of the extension officers. And then we'll work with the farmers very closely, and we enjoy like that. We visit the farmers, okay? And we work with, this is a farmer. She's very, very efficient and she is very capable. And now she started with like 4 or 5 cows. Now she has like 100 milking cows, and has her son owns, another farm, for example. Okay? So feeding interventions are very important. Feeding system modified, adapted for use by the farmer are very important. I introduce you to the food feed system that I have modified from the work of Professor Devendra. Here again, he is gone also. The late Devendra from Malaysia. Devendra has published a lot on feed resources. So unfortunately, he's gone, okay? So I learned a lot from him. We work together. We're good friends, my senior friends, okay? But I was much honored by him and by others. So I am very lucky to learn from them. Now it's my time to transfer into you guys, the young people, okay? To listen, to read more, and to generate more effective work, okay? Like that. Food feed system is the benefit by mixing together of the feeds that can be used by the humans and the crop residue or the byproducts by the animals. Like the crop residue from leguminous crops can be utilized. For example, this is one, like in a small plot of land, the farmers can grow different kind of fodder trees. Okay, here I have raised to you 3 kinds of fodder trees. Okay, one around the fence here, another one around another fence. Okay, like Lucina, we have a lot there. Okay, second one is like Firmingia. This is another leguminous fodder. You can choose another one like Sesbania, okay, and Moringa, for example. We have it's there. You have to employ them. You have to use them. You have to exploit them around the corner of the farmers here. Used as feed. Now, the leaf can be vegetables for human, and also if you have a lot for animals, okay, and they fix nitrogen to the soil. And in the lowland there, you can grow grass, right? There are two kinds of grass, right? Like Professor Novi, you can see that there are two kinds, the climbing like signal grass, rooted grass, and the erected type grass, napier grass, guinea grass, things like that. Napier erected type should be modified, should be recommended, because they can produce more additional biomass to the climbing type grass, in a small plot of land. So the standing erected type grass should be introduced and recommended. And farmers can cut carry. They can manage. We train them in, they can manage. They've been doing that. So the two kinds. Now onward to another crop, cash crop. Cassava is grown there, but we have to we should mix with leguminous, any kinds of legumes that can be introduced. This one you can be flexible. They could harvest it, different kind of mixture, grass and legumes here. You can do. You don't need a fancy type of legumes. It's the local legumes you have that should deserve more attention. Okay, here is a Firmingia. Firmingia can be harvested like this. After 4 months you can cut, and then the regrowth can be harvested and sun dry or make silage like that. It's rich in protein and phytonutrient. Okay, there's our friend from U of Sydney, professor from U of Sydney, who came to visit us. So this is just only a few examples, but there are more examples, in Indonesia that should be introduced. Here, this is by Indonesia, Dr. Jayanegara et al. Here from Bogor University, right? Bogor Ag University. He showed very nice different kinds of fodder trees, shrubs that especially very rich in protein, over here, and other intermediate level of fiber, especially the protein and the phytonutrient that I didn't have the data here, but this is just examples of the fodder tree shrub available in any country. Okay, in any country can be done. Okay, now phytonutrient can be used now by what? Because of the global warming, we need to reduce the methane in the rumen. Phytonutrients have been demonstrated to reduce methane production in the rumen. So we have been working on using combination of phytonutrients from not the plant, but from the waste, from the fruit waste, from the fruit peels waste, like mangosteen peel. You have mangosteen, you have banana, you have rambutan, right? You have many kinds of fruit there. So if we study, investigate, and see concentration of phytonutrients, particularly the condensed tannins and the saponins, they are very effective in utilizing this by the mode of action in the rumen. Okay, they will work with, that will interfere with the growth, with the expansion of protozoa and methanogens. Okay, they can reduce or destroy or block the transfer of the ions, nutrient ions, or protozoa, where protozoa are the place where methanogen live or adhere to. So by using supplementation of this fruit peel pellet, we were able to reduce the number of protozoa and the methanogens, okay, as well as the volume of methane produced in the rumen. So this is one of the examples of the work. Okay, you can see how they can interfere and how they can produce the mode of action. Okay, and there have been a lot of work published in this area, but I would recommend that. I think that the most important is to utilize whatever is available there, okay, and that could be simply prepared, modified, and use it, in a practical way. It is the most important. Some of them are very difficult to prepare, and although they have, effectiveness. Okay, you can see this one resources is how phytonutrient can impact on different digestive tract. Okay, you can go ahead later on and explore more details of this. Let me just have a sip of water. To further show you different sources, you can see here: mangosteen, banana peel, raw banana, dragon fruit peel, rambutan, and pots of leguminous shrub trees can be sesbania, so on and so forth. Okay. From the research work, please go on and visit our publication from like the last 10 years. We have published in many journals. You can see effect of this kind of starting from powder like that, and then later on form them into a pellet that they very beautifully impact on the rumen fermentation end products, especially improving C3 and reducing the methane. Okay, please search and then review more data from the papers. Many papers you can see under Wanapat. Okay, you can see a lot of work there for your information. Currently, there's a product that we developed in a pellet like that. We call bar flop, and the latest one is Marabac, which is just recently published. Okay, you can see this Marabac here is a combination of peels from mangosteen fruit peel, banana peel, and also what's another one is anyway, it's a fruit peel. Okay, fruit peel, three or four kinds. You will see later on that will impact on the rumen fermentation. But one important finding that we have observed, that we have obtained, was we have found that Fibrobacter succinogenes, one of the cellulite bacteria, which is Fibrobacter succinogenes among the 3 species, that has been significantly increased through the supplementation of Marabac. And the importance or significance of increase was that Fibrobacter succinogenes here is the non-hydrogen producing bacteria. So which means that, they don't produce hydrogen. Okay, they trap the hydrogen and they produce more C3 at the same time. So which is different among the other 2 cellulite bacteria. So with this, we now are working on it more. What happened, okay, in terms of effect on Fibrobacter succinogene. Okay, this one also effect of by the former PhD student from Cambodia that published this paper in terms of level of supplementation. Okay, on the rumen pH, you see of the pellet. This is supplement of the pellet on the pH. Okay, that you can go ahead and do that again. And some work, former student, PhD student also work on this effect of different comparing mangosteen peel powder and banana powder and the combination. You could see that the combination and the 2 sources and the combination were significantly different or improved as compared to the control treatment. Okay, so please go on and see this one. in terms of milk yield, also significantly improved by us. Now I got it. Rambutan. Rambutan fruit peel, okay? We eat it, right? You eat it every day. It's there. Rambutan is from the canning factory. So these peels were available from the canning factory, okay? Distributed out, located throughout the country, especially in the northern part of Thailand. You see many fruit peels from this factory that we have been able to collect and or bought from them as well. Okay, you can see, and again, many of these are available in Indonesia. So please and explore more. Okay, this is this work was published in Animals. You can see this work. Okay, again, and I move a little bit faster. Professor Novi Mayasari, I still have time. I still have time. Okay, okay. I'm telling myself, not asking. Okay, so anyway, so combination of the supplementation of 3 treatment also improve the fermentation. Okay, may I just move on? Okay, reduce protozoa, increase proteolytic bacteria. May I move a little bit faster? Okay, now many work, in vivo work have been collected and reported. You can see here, Marabac, dragon fruit peel, mangosteen. You can see, you please search and review this effect of strategic supplementation. Only small amount of pellet was used, not aiming at a lot of supplementation because it could be expensive. Okay. So strategic supplement. To the graduate student who are doing some work, you can share. Some of you may have done some work here. We can share and see, and then go further. Now we are at the stage of employing this one at industrial level to see how possible it could be. At the same time, we plan to do more supplementation of Marabac with more of the dairy cows and fattening beef cattle to see this effect on the quality of the product, as I have mentioned, particularly the bioactive compounds like peptides and conjugated linoleic acid would be significantly improved or not, and to what extent. Okay, this need to be done. Okay, now may I just mention that the current interest in terms of research is in the area of nutrigenomics. Okay, nutrigenomics is very important. Okay, in Indonesia, some of our friends there also have been interested to see the nutrient affecting on the cellular level and how they can activate the mechanisms with the utilization of the nutrient, particularly the transportation of the nutrient across the cell, right, for further synthesis of the product. With this, I myself have been interested, and I have to learn, and I have provided some opportunities for a young student, right? I have just nominated one to work with professor at Oregon State University in the U.S. He just went there 2 weeks ago. Professor Bionas here. The name is here. Bionas et al. Okay, at Oregon State University. He just sent me the message that, Professor, I also have Indonesian friend in the same lab. Just this morning. I said, great, you talk to each other a lot. And he said that this student also know Professor very well. Oh, even better. This Indonesian student, they're working together there at Oregon State. He went there and now he's learning about this, how phytonutrient could affect on the activators of the peroxisome, the PPAR, okay, peroxisome proliferation activator reactors, okay, the alpha, beta, and gamma, right, especially the beta. The beta activator, how it could effectively introduce or to intrigue the mechanism, right? This one, Professor Biomass proposed the effect of the activators, PPAR here, okay, on the overall systems of fatty acid in dairy cattle. So, you can see the interrelationship of the parameters of the activators over here, okay, and see the details how he proposed this from the literature, okay. So, well. So among many resources of meat, okay, Professor Novi, you can see that there are many kinds of meat that have been addressed, especially the lab-grown meat, okay, the culture meat, the plant-based protein source, and then the insect-based protein. So, we have to be aware and be alert, right? We have to see what these resources are telling us about, okay? Students, young students, you have to be alert and see. As compared to the expansion and the development of a genuine livestock production, okay, we need to learn, we need to be aware how they come about, how they can come about, and how comparable. But to tell you that, okay, this culture or lab meat is from Professor Mark Post, right, who's a professor over there in the Netherlands, right, at the Maastricht University in the Netherlands. He started this one, the culture meat. You can see, and we need to learn and see how it worked, right? This lab-grown meat is paramount. It's important, but the cost is still very, very expensive. So just a little bit there, you can see how the biopsy has been done. You can see that the stem cell can be taken and can be enlarged, can be expanded, okay, into more meat through the use of the bioreactor. You can see into the product like that. Okay,
pardon me, Professor Wanapat, pardon me.
Yes.
How many slides do you have still?
I will be fast. I should be like in not more than 10 minutes.
Okay.
Actually, I have spare my time with you today and to talk with the staff member as well. But it could be a little bit later than scheduled. Is that okay?
Yeah.
I'll be fast. I'll try my best. Okay.
Yeah. Okay. The thing is, we still have the agenda after the discussion for internal discussion. So, is it possible to manage your slide within 5 minutes? Is it
I will.
okay?
Here we go. We can talk about this later on. You can see this kind of plant-based meeting, and here is the insects protein. It's a very hot issue. Bioscience Journal is hosting the webinar on the 28th of September. I will be the first speaker to talk about the insects, okay, on the 28th of this month, okay? So that's why I said I've been very busy. So you can see this insect work, okay? Professor from Bogor Act University will also be there. We can see that. All right, now, networking is very important. So this is the good start, okay? So we should continue our connection together. You will see, here we go. I have been with some friends. Okay, I will be a little fast here to show you that this is just 2 years ago with the honor from the Chinese government to provide the Friendship Award that I was there. Okay, I just move a little bit fast. I'm not going to show you this one. It's in Chinese, but it's too long. Maybe just skip this one. Okay, some pictures with the yak. This is another potential ruminant in China, in Mongolia. In Mongolia. Very important, very interesting that we should learn more. So I've been lucky, I said that I've been lucky to travel around like this one in Myanmar, okay, to help with the government there for the livestock development. Pakistan, okay, the buffalo work. So some friends from Indonesia also were there. So we know each other, okay. This is the World Buffalo Congress that is organized every 5 years. The young people can look up to and present So some of the long time work, okay, here is university, Professor Cabriep, University of California, Davis, Professor Yu from Ohio State. I sent some students there, long time. So these young students, they have graduated already. So Professor Max Sweeney from Australia. So this is why, the benefit of working together will really come and enrich your experience and work. Here's the University of Nottingham work, the dairy farming, dairy industrial forum work is very important. Here, most important is Indonesia. So many friends from Indonesia, you can see here, it's in Yogyakarta, this one, I think, Yogyakarta, yes.
UGM, Prof. UGM, Prof.
Yes, okay.
Dr. Budi.
Yes. And many other places, okay, that I can, so many places that we can do, just like this one. Yeah. Think globally, act locally, okay? Think globally together, connect with each other internationally, act locally. You guys are going to work there in Indonesia. So that with all hands together, we will be very efficient, okay, to work together. So in conclusion, okay, animal food protein or from animal production is very important, very essential to the production, economically, to the well-being of the people, and especially through the connections of the people together, not only nationally, but internationally. Okay, this means a lot to the stakeholders. All stakeholders in the food chain together, okay, are the key person to produce the food, good quality food for the well-being of the people. Okay, but we are animal scientists. We are the technologists. Okay, and we are helping to serve and to transform the technology to the young people. The young people should be understandable and should work, okay, happily, work hard, but happy with your work to produce more efficient work with livestock production, okay? And I have short time here. I have introduced you the use of phytonutrients, particularly the condensed and supplements through the available food waste and the pellet that could be, introduced and intervened to be used as strategic supplementation in the feeding regimes. But more work needs to be investigated, okay, for getting more data on production and quality of the work. Okay, but lastly, I like to urge you and motivate all of the people, especially young people here, to work together. Okay, to work together, not only for yourself, but to improve the stakeholder, the society, the farmers as a whole, in order for them to gain more economically and socially with their well-being. So today, it's my great honor again. I wish to express my sincere thanks to Budget Jaran Universitas, through dean and staff members, okay, to the international office, Mr. Setyawan, okay, and Pusmobi, Makasari, and everybody, especially participants here. I see this is the first time through a Zoom virtual lecture that is like 400 participants. It is the highest I've ever done, right? But through the conference, okay, in person that's more, like 1,000. But through the Zoom meeting like this, you guys have done a great job already. I really appreciate it, and I enjoy talking to you guys. And this is the beginning. Please continue our dialogue and keep connecting me for anything that you think that I will be useful for your work, okay, and to help the farmers. Thank you for the opportunity. Terima kasih. Sawadee-kap.
Yeah, thank you so much, Professor Wanapat. It was really amazing presentation. I was a bit hesitant to stop you because I really want to continue, but the committee remind me that we will have another agenda. So I will remind you after the discussion, there will be internal discussion to talk about the future collaboration.
I am personally sorry. I'm personally sorry for the late presentation of buffalo, especially the fibrobacter succinogenes. And on top of that, we have found less methane production have been generated in the rumen of buffaloes. Okay, but with the impact or effectiveness of using phytonutrients here, I think that they should be similar in their mode of action in any ruminants, because condensed tannin and saponin, for example, have been utilized here, that they would then they would impact on the protozoa firstly, okay, as I have mentioned, okay, in terms of interfering with the transport of the nutrient ions through their cell, and they would be destroyed or reduced in number of protozoa, whereby the methanogens adhered to the membranes there would be also reduced. Hence, the volume methane production would be subsequently reduced in the rumen. Secondly, the effect of phytonutrients like saponins, the contaminants will also support the would also impact on the fiber by coating on the particle of the fibers, which will render difficulties for other bacteria, or secretion of enzymes will be more difficult to degrade the substrates, especially the fiber, whereby less hydrogen will be reduced via the production of acetate. Okay, so less hydrogen will be produced. Okay, less methane synthesis will be then reduced. So the mode of action will be similar, but the extent of the sources in different species may be different. The extent of the sources may be different. As I said, in swamp buffalo could be more effective, more pronounced than other species.
Okay, thank you for the answer. Okay, next question is from Evander. It's also from class E. Is there any technology or feed material for ruminant that can reduce expenditure of methane in rumen? I think you already explained, but probably you can repeat again. If there is the technology, how that
thank you. Terima kasih for the question. Now we have to think back that methane is produced in the rumen, okay, through methanogens, right? Through the available substrates of carbon dioxide and hydrogen, right? Produced in the rumen, okay. So any feed that can contain phytonutrients. This is an example of the substrates that could be more effective in reducing the methane, right? Like phytonutrient, like condensed tannin, saponin, and other substrate and other compounds that could be available as well. But this is an example of that. But to reduce methane, it means that the efficiency was less. In order to be more efficient in production, if we can reduce the methane, okay, by blocking the synthesis of methane through carbon and hydrogen, as I showed you already, that the hydrogen here will be trapped and then synthesized into C3, propionic acid, which is a glucogenic compound that will be C3 will be absorbed through rumen wall and transported to the liver, and in the liver it will be metabolized through gluconeogenesis, right? And glucose hence will be produced, and glucose is very important, their substrate for the production of in the systems of the ruminants, where energy source like ATP will be generated, and glucose substrate as such will be utilized in the synthesis of carbohydrate or sugar in particularly in lactating animals, right? So if the feeds contain these compounds and could produce more efficiency for the fermentation activities, then you will be able to reduce the methane volume production as such. Okay, a good example has shown you phytonutrients would be one, among other other sources. So look for feed resources that contain phytonutrients. I have given you some examples like cassava, zucchini, cisbania, moringa, so on and so forth. They contain phytonutrients.
Yeah.
Okay?
Yeah. Okay, thank you. The next question is also the same question actually. So I'm gonna move to the last question because of the time. We have to finish in about 2 minutes. But let's see. The last question, maybe you can just answer it very briefly. So the question from Dela. So the raw fish or the forage is very important for ruminant, of course. But the lack of the land is the problem. How we provide the forage? Is there any way, or is there any solution? How actually to answer the problem?
Good question. I have already given you a good example. You have to introduce the food feed system. Okay, is one thing. Okay, provide more diversified resources of feed on the farms there. Secondly, is also to open up for using other resources from open areas, from public areas, where the farmers can have an access to and cut and carry into the system. But we need to provide the training to the farmers to establish their own plots, but small plots that with more biomass. More harvesting throughout the season is most important, especially harvesting at the right time with good concentration of nutrients, good quality of nutrients, especially, good protein, carbohydrate, and minerals, right? Not too much of fiber, right? So do that. And importantly is that resources locally available are there, are diversified, are multifunctional, that we need to put our efforts in introducing to the farmers, okay, to those available resources. And on top of that, introduce, the new species of resources available through the findings of research, to the finding of other recommendations from other places, okay? All together, they're combined together, then you have more biomass, more feed resource available throughout the year.
Yeah.
Terima kasih.
Terima kasih. Yeah, I think that was the last question. Yeah, I'm so sorry because I think we do have more than 25 questions from the participants, but due to the time limitation, so we cannot accommodate all the questions. So hopefully they will be away from the committee. So after the discussion session, we move on to the appreciation.